So our last speaker of the day is a clinician scientist, Dr. Jaiping Wong. He's being brought into the EHS field by Dr. Luke Kai, and he is the individual that Dr. Cave was telling you all about as opening up a new field of environmental research. And so this is going to be very exciting. I'm looking forward to hearing this, and it's research addressing pulmonary arterial hypertension. So this is a human subjects study. This is not animal model. This is real world, real life. And so these types of projects a lot of times require a lot of consideration. So this is a metallomics study.
Thank you, everybody. I'm pretty new at this, so bear with me. So today I'm a clinician. I'm a cardiac anesthesiologist. My interest is pulmonary hypertension clinically. We develop devices, ventricular assist device to help with patient with heart failure. And thank you. And one of the issues they have is the pulmonary hypertension, and pretty much all the patients after left ventricular assist device will develop pulmonary hypertension. So what my interest in this project was, which was sponsored by the P30 grant, is really to study the metal profile in pulmonary hypertension patients. I'm going to do a little background introduction. So what is a pulmonary hypertension? It's defined by the pressure of the pulmonary artery, more than 20, and there are different classes of pulmonary hypertension. Group 1 is a pulmonary arterial hypertension. So really means the artery of the pulmonary artery pressure is high. Group 2 is a pulmonary hypertension from the left heart disease. 3 is from COPD and hypoxia lung disease. Group 4 is because of pulmonary artery obstruction. Group 5 is miscellaneous. So every time when you see we approach a clinical issue, right? You have so many classifications. Pretty much means we really don't know what's going on. So that's really what it means. We know we would see that's what the cause of that. So pulmonary hypertension actually is a significant issue. The incidence is about 2 to 7.6 cases per million, and the prevalence is about 11 to 26 per million. And it's primarily a woman's disease, actually 4 times higher in women. However, if you're a man and you got a pulmonary hypertension, you do much worse. And almost half the patients have idiopathic pulmonary hypertension. That means we don't know the etiology. And the outcome actually is we're doing a little bit better. And the long-term survival has been significantly improved over the last 2 decades. The median survival is about 6 years compared to 2.8 years about 30, 40 years ago. And the 1 year survival right now is about 86 to 90%. And the reason that we got better is because we know better. We have a better awareness of pulmonary hypertension. We do have some specific PAH specific therapy. I'll talk about why that's really not ideal yet. And we put those patients on blood thinners. So these patients tend to have blood clots. And we can manage the right heart failure from the pulmonary hypertension a little bit better. And so not all pulmonary hypertension are the same. They actually have different classifications. So they have a low risk, intermediate risk, and high risk. There's another score system we use. And let's look at the pulmonary hypertension specific therapy. We treat pulmonary hypertension patient like we treat any heart failure patient. We'll put them on blood thinner, we'll give them diuretics, we'll try to get the fluid off them. And the most significant treatment so far is calcium channel blockers on pulmonary hypertension. The reason is easy to understand. If you have blocked the calcium, that's going to cause vasodilation. So once your pulmonary artery dilates, your pressure decreases. So you will have less pressure on the right heart. And nitric oxide, that's another important pathway for the pathogenesis of pulmonary hypertension. And primarily the L-arginine will be metabolized nitric oxide. Nitric oxide can work on FGC and to convert GTP to cyclin GMP. And also the cyclin GMP will be metabolized by PDE5 to GMP. So the one that's active is cyclin GMP, which is the one responsible for the vasodilation. So if this is not working, if I don't have enough cyclin GMP, the HCl will run into its really vasoconstriction and increase pressure in the pulmonary artery. And the endothelin is also an important factor too. So the endothelin will combine to endothelin A and B receptors, and both these will cause vasoconstriction. So if I want to treat a patient with pulmonary hypertension, I would use antagonists to those receptors. And the other one is probably the most common medication we use, the prostacyclin pathway. And what that will do actually will increase cyclin AMP production, which will cause vasodilation. So what are the issues with the pulmonary hypertension therapy that we have right now? There's really no treatment so far can decrease the mortality. So what will we do? We're not making them live longer. And the only one that showed a decrease in mortality was in patient actually with class 4 classification. And the average improvement on the 6-minute walk, so you let the patient walk for 6 minutes, see how far they can walk. Usually 400 meters is pretty good, and less than 160 is pretty bad. And also the mean pulmonary artery pressure. So those are the two major clinical outcomes that we look at to see how effective therapy is. It's really not that good. And for example, in the randomized clinical trials, and the average fall in PA pressure, and also it's only 6 millimeter mercury. So just to give you a general idea, the normal pulmonary artery pressure is about 20 to 30. They only drop about a 6 point. That's really not that significant. And most of them are working on the vasoconstriction. The one I show you, we want to make the pulmonary vasodilate. And actually, this is only the issue with those patients in 10% of them. So many patients, this is not the problem. And so there has been significant studies on the pathophysiology and the potential therapeutic targets on pulmonary hypertension. And you can see here, with the pulmonary hypertension, you got a lot of issues going on with the pulmonary artery. You go to the medial thickness and new edema, inflammation, thrombosis, and the plexus formation. And one of the things that I'm interested in, and Dr. Lu Chai, who's very interested in this, is really oxidative stress in pulmonary hypertension. As you can see here, through all the pulmonary hypertension study, oxidative stress has been studied extensively, and the multiple markers are elevated or affected by pulmonary hypertension. And Dr. Lu Chai, myself, we have interest in pulmonary hypertension, so we developed a mouse model for pulmonary hypertension through the VEGF receptor and antagonist SU5416 and hypoxia. So if I treat those mice with this mice will develop pulmonary hypertension. And then we use a sulforaphane, which is something you can find in the cauliflower, and what that is a really antioxidant. Our thinking was, well, once we develop the pulmonary hypertension mouse model, if we give this antioxidant sulforaphane, will that help? Actually, we did an echo on mice, and we also measured the pulmonary pressure of those mice. Actually, we were able to show that once you induce pulmonary hypertension in those mice, if we give them sulforaphane, which is antioxidant, that actually can reverse almost all the parameters for pulmonary hypertension, including the pressure, the cardiac output, and other measurements. And so we actually also did downstream analysis of those pathways, and we were able to show that actually the significant issue with oxidative stress in pulmonary hypertension in this mouse model, and certainly there are limitations in the mouse model because that's not how humans develop pulmonary hypertension. But this is probably the most commonly used mouse model. We're able to show that the multiple pathway was affected, and sulforaphane was able to reverse most of them. There was a study that was very interesting in 2019 that kind of got us interested in sort of metal and pulmonary hypertension. So this was published on PNAS. So what they were able to show, they developed a knockout mice. So in these mice, if they make those pulmonary artery smooth muscle cell, if they make those cells have R deficiency, so in those mice actually will develop pulmonary hypertension. So this is very exciting for us because this is really the first time people show you can just manipulate one iron, right? You just manipulate the iron deficiency, can develop pulmonary hypertension by itself. So that's got us interested in. So now it's really coming to the project. So sorry for all the introduction. So I was supported by the P30, Dr. Merchant, Dr. Cave, and Dr. Stace. So I was able to get funded for a small voucher award from the P30 grant. So what we want to look at is we're going to look at the pulmonary hypertension patients. The question we want to ask is there any issue with the metals? So when we talk about metals, we really think about two metals. One's heavy metals, the metal that's not good for you, and also essential metals like iron, like zinc, right? Those are good for you. So is there a correlation between those metal profiles and the pulmonary hypertension patients? And our theory was that the pulmonary hypertension was from the narrowing of the pulmonary artery, would cause increase in pressure and the right heart stress. And what many studies have shown that pulmonary hypertension got an issue with oxidative stress, right, like we showed before. And also, if you look at the metals, and has been shown that both essential metal dyshomeostasis and also heavy metals, they can all cause increased production of oxidative stress. So what we're thinking, there's correlation out there, right? Maybe they work on the same pathway. So, however, this relationship has not been clear. So with this support, we're able to study this. So we have our specific aims for the study. Really, we hypothesize that heavy metal pollution, either due to human contamination from the industrialization, urbanization, can impact everyone, especially people in the low-income communities. And we try to assess the exposure and the concentration of those environmental metals and the essential metals in three sources in the patient samples. One is whole blood, plasma, and urine samples. So this is our method. So we do have a pulmonary hypertension clinic at U of L. The director, Dr. Elkirch, he unfortunately moved to University of Nebraska. So this is a collaboration with him. So from this pulmonary hypertension clinic, we're able to get 20 patients with pulmonary hypertension. And we asked them to do a survey. They filled out the questionnaire, and also we gather urine sample, blood sample, and serum samples. So we also got 3 healthy controls as a control, so we can compare those. We collect all the blood urine samples to measure the whole blood, plasma, and urine level of 25 essential and heavy metals. So this is actually so nice about the CIEHS. They have so many core facilities. So this actually, the core facility was supported by the CIEHS. So all I do, I get the samples, I give it to them, then they measure all of them. So I really didn't need to do anything else, which I couldn't anyway. We did a statistical analysis on those. So first, let's look at the pulmonary hypertension patient characteristics. What are the 20 patients? The average age is about 57.6 years old. 80% of the patients were female. As we talked about, this is actually a female disease. And 6 patients had idiopathic pulmonary hypertension. All the patients got a pulmonary hypertension therapy. Remember, we talked about prostacyclin treatment, so everybody's on there. The mean last 6-minute walk was 312 meters. So the normal should be about 450 meters. So this is certainly a decrease. The BNP, which is an indicator of heart failure, is also increased to 118. And we did the risk scores, and among those, 4 patients are high risk, 6 are intermediate, and 9 patients were low risk. We also measured the hemodynamics. So with those patients with pulmonary hypertension, this is probably the most important thing that we do to them, is do the right heart cath. So put a catheter, a pressure catheter, in the right atrium, advance the catheter to the right ventricle, then to the PA. So we can measure the pressure in each chamber, we can measure the cardiac output. So in those patients, the right atrial pressure is 5.9 per normal, the PE pressure is 45. Normal, you and I probably got a 15 to 20. So this is just significantly elevated as expected. Cardiac output 5.1, that's relatively normal, and the PVR is 8.32. The normal should be about 1 to 2 Wood unit. And we in the questionnaire, because our interest in sulforaphane, so we asked them, is there a difference in terms of their vegetable consumption? The rationale behind this, some of the vegetables, we know they have a high level of antioxidant, some are not. Is there a correlation down there? So this is very interesting. So in all the 20 patients and the 3 controls, we did not find any significant differences in terms of sex, smoking history, and the number of exposures between the pulmonary hypertension patient and control patient. However, the control subject reported much higher consumption of sulforaphane-rich vegetables, such as broccoli, Brussels sprouts, and cauliflower. So although this is a small sample, we only have 20 patients and 3 controls, but that does show us that there could be correlation down there. So the next thing that we thought was very interesting is antimony and pulmonary hypertension. So in the pulmonary hypertension patient, the mean plasma concentration is 2.6 nanogram per ml. The mean blood concentration 1.3, the urine concentration 0.1. So all those are higher than the mean level of the 3 controls, which is 2.2, 1, and 0. Although did not reach statistical significance. One thing that there are really 2 reasons with this kind of study is, one, our patient population is very, very heterogeneous. So there's different etiology. So there might be 1 or 2 patients, this is the cause of it, that might be high, but others kind of drag it down because those are not a cause from this. So in the idiopathic pulmonary hypertension patient, we're kind of interested in those because when we say that you're idiopathic, it means we really don't know what's going on, what caused that, right? Otherwise, we would have said that. So in this, the plasma concentration is much higher, 3.1 compared to 2.5. The more interesting with the antimony was we actually found a significant correlation between the level of the antimony and the hemodynamics. So really, how bad your heart is doing. So what we found is if you have a higher level of antimony level, your mean right atrial pressure is higher. So right atrial pressure is indication of heart failure. If your pressure is high, you're going to develop heart failure. Your cardiac output is lower. The heart is pumping less with a higher level of antimony. And the cardiac index is also lower. The pulmonary vascular resistance, the offload of the right heart is increased too. And the saturation of the pulmonary artery is lower too with a higher antimony. So what does that even mean? So antimony, we did some research on that. We really didn't know what that means. So antimony is still being used to treat a parasite disease. And we know it does have a cardiac toxicity in there with a potential side effects, especially the ventricular mouth size of the guinea pig treated with very low dose antimony showed impaired contraction. So the function is decreased. And in mice, if you give antimony, can cause cardiac toxicity through induced apoptosis through the endoplasmic reticulum stress. And there is a significant, what we found is a significant correlation between the antimony level and the hemodynamics of pulmonary artery. So this has not been reported. So we still try to figure out what that means, what's our next step going to be. So there might potential to be an environmental antimony level with the pulmonary hypertension and right heart dysfunction. So we certainly want more study on this. So we analyzed 25 metals. So we found some other thing that we thought that was very interesting. The other, one of the things that there was discussion regarding the cadmium, and we actually found a very similar thing. So we looked at the cadmium level in pulmonary hypertension, blood, plasma, and urine. What was striking is in the urine of the pulmonary hypertension patient, their cadmium level is about 10 times more than the control. And also in the blood, there's about 6-7 times more down here. So our hypothesis is, could this be the cause? Could be one of the cause, right? Probably not all the cause. Could this be the cause of it? And we also found some other metal we thought it was interesting. We found a manganese level is higher in the pulmonary hypertension model. Patients, especially in the urine, is significant, probably 20 times higher. Lead level is higher in pulmonary hypertension, and we all know lead can cause significant issues with the lungs and the hearts, and we found the level in the blood is significantly higher. It's 12 versus about 5. And also interesting in the zinc level. When we look at the zinc level, the blood and the plasma level is about the same, but the urine level actually is higher. So I had a discussion with Dr. Lu Cai, and he's the expert on this. So he said that could be indicating you're losing zinc, although your level is still the same, but that could indicate you're actually losing zinc through whatever mechanism in those patients. Arsenic. So what we also found that arsenic level was higher in the urine compared to the control group. So to summarize, what we saw on the metals in pulmonary hypertension, we compared to the controls. The pulmonary hypertension patient had a lower blood concentration of thorium, and we started trying to figure it out what that means, what could be the potential mechanisms on there. The cadmium was 5 times higher, and the lead level was twice as high in the pulmonary hypertension patient, although they did not reach statistical significance on that. The plasma level of sodium was higher, lead was twice as high, and manganese and arsenic level is also higher. All the, we have multiple metals, although they're not statistically significant. However, the urine metal concentration of arsenic, cadmium, and zinc are significantly higher than the patient with pulmonary hypertension. So from this, so we look at this, we see what can we do from this data, right? We certainly give us some hints, and what do we take from here? So one of the project that came out of this, really through the discussion with Dr. Cave and his leadership. So we see, well, why don't we look at how cadmium? Because cadmium is significantly higher in the urine, right? It's 10 times. How this affect the pulmonary hypertension remodeling and RV dysfunction? So we actually was able to get another grant from the CIEHS to support this study. So we already have a pulmonary hypertension mouse model. So what we see, what if we feed those mice with cadmium with a different dosage? So we have a low dose, intermediate dose, and high dose, and also we feed them at different time before we induce pulmonary hypertension, during and after. What we want to see is, how does the cadmium affect this at all, or in what ways, in what dosage, and in what time and period would that be significant? So this is one project. We actually submitted R21 to INH. We'll see. So the second one, this actually was definitely was led by Dr. Matthew Keefe and Dr. Stace to do a multi-center study. And so our interest was, well, we think there's something wrong with the metal with the pulmonary hypertension patients. And how do we study this? One of the things that we came up with, well, why don't we look at the lung transplant patients? So they're going to take the lung out, right? The old lung will be out. Nobody wants it. We can take it. We can analyze it. All those lung transplant patients, by definition, they got a pulmonary hypertension. I would say 95%, right? That's the reason that they got a new lung, because their lung's not working. So we actually work with Emory P30. So it's a P30 interaction led by Dr. Cave. And so what we try to look at is, we also work at UNMC, University of Nebraska. So we want to get the tissues, the explanted lung, and also get a piece of pulmonary artery. So we're going to analyze the metal levels, all the 25. We already did that, and to see if there's a difference, right? Maybe compared to normal, maybe there's a significantly higher. The other thing we really want to do is also that we want to look at the distribution of those metals on those tissues. Right now, I don't think our center can do that yet. If you just got a tissue slice, you can just analyze, where's the metal, maybe it's in the parenchyma, in the small vessel, all those, and that will be our long-term goal. In conclusion, it seems like the abnormalities in essential and heavy metal concentrations in pulmonary hypertension patients may potentially act as a risk factor for the development or acceleration of pulmonary hypertension pathophysiology. And the potential metal effects, the bad effects of metals, might be worsened by the lower consumption of antioxidant vegetables. If we find that, we might suggest that those patients probably should eat more vegetables or take supplements. And defining the potential factors in the development of pulmonary hypertension with the environmental factors are certainly needed. Thank you very much.